Hello and welcome to everyone joining us tonight virtually for Indian River Lakes Conservancy's Summer Science Series program. My name is Heidi Sauerwein and I'm the program coordinator with the Indian River Lakes Conservancy. And we are all missing you in person tonight. As you can see on your screen, this is where we normally hold our Summer Science Series programs. So we hope you can join us here next summer if you haven't already visited us at our Trailside Learning Center at the Redwood Hill Preserve. But thank you to Rodger for being flexible and offering to do this program virtually tonight. While everyone's typing in the chat box where you're from, I'm just going to say a few words about the Indian River Lakes Conservancy. We are a donor-supported land trust dedicated to preserving the unique natural character of the Indian River Lakes region of northern New York State. And I have a couple of little maps if you're from out of the area or unfamiliar with the Conservancy. So this is our watershed, and we're located mostly in the county that's highlighted there on your little New York State map. Our mission is fourfold. It encompasses protection, education, recreation, and we want to do this forever in perpetuity. So we're taking the long range view on things. Tonight's presenter is Rodger Klindt. Rodger is from the New York State Department of Environmental Conservation. A little bit more on Rodger in a second. We also hope you can join us for our upcoming presentation, Kate Cleary. She's a professor at St. Lawrence University, and she's going to be talking about genetics and movements of wildlife populations. You can ask a question by using your chat box, or at the end of the presentation, you'll also have an opportunity to unmute yourself and ask Rodger a question as well. So we hope that you will take advantage of being able to speak with Rodger. So a little bit more about Rodger. He's been working with the DEC starting in 1983 as a summer intern. That was the year I was born. As a summer intern from SUNY ESF in Syracuse. He worked seasonally for several years with a hiatus where he studied muskellunge along the St. Lawrence River, and he became a permanent DEC employee in 1990. And since then, he has worked all over the 5-county region, from studying Adirondack trout ponds all the way to Lake Ontario. The last 15 years have been focused on warm water lakes and rivers, as well as the St. Lawrence. And he's worked on lake sturgeon recovery efforts since the early 1990s, and he's seen a lot of progress over that time. So without further ado, thank you, Rodger, for joining us. I'll let you take it away.
Okay, well, we'll see if I get this all figured out, and if I need help, I'll call.
Yeah, I'll be here.
Let's see. All right, hopefully you see a sturgeon up there.
There it is.
All right. Get started. Okay, so normally if I do a presentation, I'm in a room filled with people, and I really enjoy the interaction with folks. So this is going to be a little odd for me. This is the first time I've ever done a virtual presentation. So bear with me if I hang up somewhere along the line. I'll try to regroup, and hopefully it'll all make sense in the end. So I'm going to talk about lake sturgeon recovery. As it relates to the Indian River Lakes, towards the end of this, we'll get into Black Lake specifically to bring it home, you know, why I'm here. But just to make note of that first picture you see of the sturgeon, that was a watercolor done by folks from the original Biological Survey of New York State, which was in the late 1920s and through the 1930s. And this original artwork is amazing. And it was literally rescued from a dumpster. It got stashed away in a closet. Somebody said, clean that place out. And someone luckily recognized what was hanging out of the dumpster and saved these illustrations that were made. And they're on our website if anybody's curious about more of them. It's really amazing work. So let's move on. So what are sturgeon and why should we care? So number one, these fish are living dinosaurs. They basically remain unchanged for the last 200 million years. We do have a few other dinosaur type fish in our region, long-nosed gar, bowfin, also known as dogfish, are also some of that ancient lineage of fishes that really hasn't changed much in many, many millions of years. Sturgeon are indicators of quality environments. So even though things may look bad out there at times, if these sturgeon are surviving there, it may look bad, but it's not as bad as you think. Trust me, the places that are really bad, sturgeon will not exist. And last and probably most important, they are wicked cool fish. So we got 3 species of sturgeon. You know, the last fish on there isn't a sturgeon, obviously, but I'll talk about him too. The short-nosed sturgeon is basically along the Atlantic seaboard. So it exists in brackish water, freshwater, the Hudson River. It is state listed as endangered. It doesn't get very big. You know, 3.5 to 4 feet is about as big as that fish gets. So tonight we're talking about lake sturgeon, which inhabit the Great Lakes drainage, some of the upper Mississippi, even down into the Ohio. It's got a pretty broad range, but the Great Lakes is the main focus for the lake sturgeon. And it is listed as a threatened species for New York State. Lastly, the Atlantic sturgeon, you know, lives pretty much by its name. It's essentially a marine fish, comes into fresh waters to spawn, not unlike salmon. It's federally listed in different ways. It's listed by genetic groups and where they spawn. So most of them, I think, are listed as endangered. I think there's one spawning aggregation that's listed as threatened. But these are the big fish. These fish can grow upwards of 1,000 pounds. And actually the population utilizing the Hudson River to spawn, also known as the New York bite, is one of the healthiest populations, I believe, along the Atlantic seaboard. Now the paddlefish on the bottom is native to our Allegheny area, so it's the very southwestern part of New York State. And we've also done some recovery efforts with that fish. And the reason it's on here is because it's again one of those ancient fishes that really hasn't changed much. It has more in common physically with sharks than it does with modern bony fishes. So sturgeon of the world. There's about 25 species of sturgeon worldwide, and I say about for a good reason. These fish are depressed. In a lot of cases, we don't know if some of these species are extinct or not. Every once in a while, there will be a news article, one fish just ready to go on the extinction list, and lo and behold, somebody catches one. So there's such low density that people haven't, in some cases, haven't seen them for decades. So we'll say that there's about 25 species. It could be a little bit either way, but that's what you've got worldwide. And through history, we've had a few different commercial uses. Obviously, it's a large fish. You know, there's a lot of meat, a lot of protein for consumption. Caviar, which is basically just pickled fish eggs, was very popular. I'm sure it is still very popular now. They use a lot of other fish other than just sturgeon. And isinglass was a clarifying agent used for the wine industry, but basically they've come up with other things to use for that process. So as far as today's commercial uses for sturgeon, you have meat and caviar, pretty much it. But regardless, Every species of sturgeon is imperiled in some way or another. There's no place where there are just tons of sturgeon like they used to be historically. That just doesn't exist. So Lake Sturgeon Bio 101, what do we want to know about these fish? They're long-lived. They can live upwards of 100 years. I think most of the fish that we've handled that we've been able to age top out at really no more than 40 years of age. You know, if you see the 100-year-old fish, that is the, you know, the rare, the extreme, you know, highly unusual. They can get quite large, upwards of 7 feet and 300 pounds. Every time, you know, somebody comes up and says, I've seen like an 8 or 10 foot fish, they've probably seen a lake sturgeon, but it was probably not 8 or 10 feet. The largest one actually ever recorded was a little over 7 feet long and weighed a little over 300 pounds. Is there a bigger fish out there somewhere? Maybe. But by and large, those of us who look to records, we haven't seen it. They're benthic feeders. That means they eat stuff off the bottom. So invertebrates, mussels, small fish, basically anything that doesn't get out of the way. These things are like vacuum cleaners. And if it doesn't get out of the way, it goes down the pipe. Interesting thing about mussels, when I say mussels, I mean any of the clams that grow, the native clams, if they're small enough, sturgeon will eat them. But also when zebra mussels came into our systems, that became a new food source for lake sturgeon. The same thing with round gobies. Round gobies are kind of unique in that they tend to defend their territory. So you've got a 3-inch fish defending itself against you know, something 5 feet long, and it's going to go down the vacuum cleaner hose. So now we get into some of the aspects of sturgeon which sort of don't work well with modern times. So the females generally don't reach spawning age until they're 20 to 30 years old. Now maybe we've pushed that back with some of our really fast growing places like Black Lake and Oneida Lake, which maybe we've seen some females mature as early as 15. But when you think about most fishes, they're going to mature and be able to reproduce between the ages of, you know, 3 and 4 years of age. But this fish doesn't even have a chance until it's got 15, 20, 30 years under its belt. So that's sort of a detriment in today's age. Maybe back 200 million years ago, that life strategy worked out pretty well, but it's a little challenging for them now. And to top that off, the females only spawn every 3 to 5 years. So again, like most fish, once they reach sexual maturity, they can spawn every year. A female takes 3 to 5 years for those eggs to mature. It's a very long process. So if you can imagine, if these fish are harvested and taken out of the system, they can't replenish themselves nearly as quickly as most other species can. And that will come up later. Migratory, they move upstream for spawning. So they need swift water. They want well oxygenated water moving over their eggs on clean substrate. So impediments to migration have been a problem for sturgeon. And again, they need high quality habitat, well oxygenated water, and they spawn over clean gravel that's not impacted with sediment or other waste products. So looking at the fish, the head end, most interesting part, you notice those 4 barbels that hang down. Those barbels, those whiskers, have the same purpose as the catfish in bullheads. Those are sensory receptors. Those are for sensing chemical signals that something is alive under my mouth and I need to eat it. Okay, so that's, you know, given where their mouth is, which is just under the eye, it's on the bottom of the fish, it allows that vacuum cleaner hose to come out. It makes a lot of sense for where they're positioned and what they do. Now, all those little pits that you see around the nose, and in the next picture we'll show them again, are very similar to what sharks have. They're called ampullae of Lorenzini, and those are electrical receptors. So sharks use them for feeding. I think most advantageously at night, like the reef sharks feed at night, and they obviously they don't be able to see as well. So they can use that receptor system to help them find prey. Well, similarly, the lake sturgeon is using both its barbels chemically and those little pits electrically to find things to eat. And right there is the mouth. And the mouth is very muscular. It can protrude a little bit like a vacuum cleaner hose, a few inches. There's no teeth in there. So basically that muscular mouth can just grab and grind. And when they swallow food, the first place they go, and I don't know the name of the organ, and I apologize for that, but they've got a very muscular organ at the top of their digestive system. It's very much like a chicken gizzard. You know, it's just there to crush and squeeze anything that they've taken in. So they're very efficient at finding and consuming food. Of note here, you see that individual is holding that juvenile sturgeon very gingerly. And the reason for that are the bony scutes along the top, the side, and the bottom of the fish. And they're very razor sharp, they're hook-like. Through the years, anybody who's handled small fish has gotten some pretty nasty cuts from these things. It's a wonderful armor system that they've got for protection. As they age, typically those will wear down, and we'll see that in a moment. But juvenile sturgeon will be sort of a light chocolate brown with black blotches on them. So this is an example of what those bony scutes look like with an adult fish. You'll notice you can just barely see the line. They're just remnants of those bony plates, and the rest of that is just skin. And there's different opinions about why they wear away or why they disappear. I think in this particular photograph, for anybody who is familiar with lamprey eels, they're not really eels, we'll call them lamprey eels though, because most people know them that way. They are parasites and they attach to the sides of fish. And if you look right in the center, you'll see clean tooth marks from a lamprey and see all those scratches on the fish where the fish was basically rubbing it off on the rocks. So the fish do a lot of abrasive cleaning when those parasites get on. So again, here's the basic distribution in the US. I couldn't find a good photograph or a good illustration of the entire distribution. You see, focuses on the Great Lakes, down into the upper Mississippi, Ohio, Tennessee rivers, and as far as Canada goes all the way up to James Bay, out into Quebec, which is where it's still within the Great Lakes drainage, draining to the St. Lawrence River, and to the northwest out into Manitoba. So they have pretty broad range in North America. So sturgeon issues, there's a few. So that's the bottom side of the Moses Saunders Dam in Messina. And dams do a couple of things. Number one, remember sturgeon are migratory for spawning. So obviously that concrete wall is a barrier to them for moving to where they can find adequate spawning. The other thing is you build dams right on their spawning grounds. You want that swift water. That's whether the dam is for milling, for hydroelectric, but you want that swift water. And with that swift water comes the clean substrate, the gravel, cobble that the sturgeon are looking for. So dams sort of have a double whammy, a couple strikes against sturgeon. And it's not just lake sturgeon, it's all the other species as well. Over harvest. So it really wasn't until the 1960s that people began to understand the biology of the sturgeon and think about what harvest means. And, you know, back in at the turn of the, you know, previous century, it was just food. You didn't think about a conservative effort for animals like this. And the harvest records were just amazing, but with the tonnage of fish that were harvested in all the Great Lakes. And once they began their decline, it was a precipitous descent. So remember, these fish can't repopulate themselves very quickly. It takes, you know, decades and decades to start making a difference. So over harvest, you know, taking fish out of the system was a big issue. Pollution, obviously they need quality environments. And this picture is here for a reason. The timber industry was very big in upstate New York and in all of New York, really. And, you know, not only were logs habitually floated downstream where they could, you know, scour and damage spawning areas, also pulp waste, bark waste, sawdust, that stuff does not decompose quickly. So even in the Black River, where I've done a fair amount of work from all of the timber and pulp industry, I can go down into the Delta on Lake Ontario and still find remnants of you know, wood waste that's probably been there for 50 or 60 years. It looks just like it could have been there, you know, a few weeks ago. So pollution is not just chemicals and things dumped in the water. It can be other things as well. Anything that can foul the spawning beds. And lastly, disease. If folks may recall, about 2005, we got this nasty viral disease in the Great Lakes system called VHS, viral hemorrhagic septicemia. And it really did a number on muskellunge, adult muskellunge in the St. Lawrence River. It was very sad to see all those beautiful big fish dead. Interesting thing about sturgeon was they knew, as the researchers, they knew we were handling a fair number of fish, so they asked us to get blood samples for them. And we sent stuff out, and what came back was apparently these fish, or what they refer to as refractory, they can't get VHS because those protein receptors on that virus are much newer than this fish. This fish is basically 200 million years old, so the virus really couldn't attack to it. But what they are susceptible to is botulism E. And botulism has been around forever. Over the last 20 years or so, it rears its ugly head in the Great Lakes. A lot of that we think is because of zebra mussels. Zebra and quagga mussels, they set up this anaerobic system under certain conditions, and the bacteria produces the toxin, and it goes right up the food chain. So not only will sturgeon eat zebra mussels, they also eat gobies, which are right there on the zebra mussel beds. So botulism E is probably about the worst thing going for lake sturgeon in our Great Lakes system right now. So, like Heidi said, we've been working with sturgeon since the early 90s, doing our thing, you know, whatever we could. But, you know, a couple of years ago, we finally put a plan together. What's it going to take to get this thing delisted, you know, not on our threatened species list anymore? So we have the recovery plan, which is, you know, our action plan. If anybody's interested, it's on the website, on our DEC website. You can go and download it and peruse it. I don't know if it's all that interesting, but what we're looking for is metrics within management units, metrics, you know, numbers. So that's one of the reasons, the primary reason we were at Black Lake last September was looking to get these numbers. So the plan outlines several different things that we can do to try to build our numbers of sturgeon. One is to enhance or create spawning habitat. That's one of the big strikes against the fish. So if we can give them good places, good quality environments to spawn in. There's a benefit. Create passage around barriers, the dams. We still have a lot of dams in upstate New York. And previously, dams were basically concrete and steel. There wasn't a lot of thought for other species other than trout and salmon, which are very good swimmers. Sturgeon really are not the top of the swimming group as far as fishes go. And then lastly, artificial propagation, basically putting them, raising fish in a hatchery, which is what we've been doing since the early 90s. So creating habitat, that's where this one is going right now. When the New York side of the Moses Saunders Power Dam in Messina was relicensed, I believe that was 2003. Within the license, New York Power Authority was required to build 10 habitat improvement projects. And that ranged from turtles to fish to wetland bird species, all kinds of things. But Lake Sturgeon was one of the focuses for one of those habitat improvement projects. And so that focus was creating spawning habitat. Obviously, the dam removed spawning habitat for these fish. So the idea is to create quality habitat for them. So after a lot of research into looking for just the right place to start putting these things, contractors were hired. This is a dump barge being filled with big rock, which is anywhere from 2 to 6 inches, broken limestone. Moved by their tugboats with GPS. It's amazing the professionalism of these marine contractors and what they could do and how precise they were to put these beds, exactly as close to exact as you can imagine in water that's going over 1 meter a second, and 20 to 30 feet in depth. Just gauging where is that rock going to fall when they hit the dump levers. Pretty amazing. So here's a fly through of what happened, and I'm going to stop this once, but this is what happens when you give sturgeon what they want. Nope, and it didn't stop. That's all right. So there's other fish on this bed. Those are silver red horse suckers, which have basically the same requirements, the same timing, water temperature as lake sturgeon. So we use those fish as sort of a surrogate when we're looking for adequate areas. Say, well, if the silver red horse think it's good, then there's some reason to think that we can do something maybe with lake sturgeon and they will also find it attractive. So this was a case of when you build it, they will come. And not to get too uppity about that, we've had cases where we built it and they did not come. So this was one of our great success stories. Not all of our stories are as successful as we would hope. So moving on to fishways. Like I said, the historic typical fishway was concrete and steel baffles, and it was a straight shot, maybe several hundred feet long. And for really powerful fish, the salmon, the trout, they could navigate those quite well. But a lot of other species that still need to find migratory paths just couldn't adapt to those types of situations. So maybe 15 years or so ago, out in the Midwest, they started putting a few things together and realized when they had a flood and the river, the stream went around the dam, and you had little temporary streams that fish ran up them. So why not create something that's more like a stream, something that fish don't see as something foreign? So that's where the nature-like fishway began. And this is down at Eel Weir on the Oswegatchie River. This is the first nature-like fishway in New York State. As you can see from here, it's cut into the bedrock channel, and I'll give you another photo of it up close. It's got velocity breaks. It's got deep pools. It's got quick shoots where fish can quickly shoot through that and then rest if it wants to. Basically, these things have everything fish want. They have a mechanism to keep going forward and up at a pace that they're happy with. Within these pools, they're regular little ecosystems. We have resident fish that live there, bass, darters. You know, quite honestly, sturgeon can spawn right in that if they want to. It's really been a pretty interesting road to get to this sort of an idea of fish ways and moving fish, how they would like to be moved. So there's another one that has been built at the next impoundment up at Huvilton on the Oswegatchie, and I think this year they're going to be doing some pretty serious studies looking at how fish are moving back and forth through the system. So there's one feather in our cap for fish passage. We're getting, at least we think we're getting better at it, and this could definitely benefit our sturgeon. So lastly, we get to raising fish in a hatchery, and we've been doing this since the 1860s, I believe. Seth Green, Rochester native, is known as the father of modern fish culture. He is the one who basically started these techniques that we still use today with little modification. But we go out, we catch mature, ready to spawn fish. We squeeze eggs in a bowl. We squeeze sperm in a bowl, and voila, we have fertilized eggs, and they go to the hatchery. Well, that's great if you have populations of fish where you can just go and grab actively spawning fish. Unfortunately, with lake sturgeon, we don't have that in New York State. Remember, they're threatened. They're sort of a low density fish. So we've figured this out over years, you know, not on our own. Other folks have been doing it, but we go out and we collect fish. Our largest population is right below the Moses Saunders Power Dam. So within the South Channel, which is the bypass region, it used to be the Long Sioux Rapids. It's basically still water, and fish go there to stage, getting ready for spawning, because the water typically warms up a little bit more. So that gives them an opportunity for their eggs and sperm to mature, you know, in warmer water before they're ready to go out into the main current and try to find a spawning spot. So we go out and we collect fish in these really large mesh gill nets, and about the only thing that we can catch other than sturgeon are a few carp. I think in all the years I've done this, we've caught one muskellunge, which we were able to release alive. But we will, you know, run a net and hopefully fill a tub of fish. So you might say, oh, well, look at all those fish you got. No problem. How hard could it be to get fish that, you know, you can get eggs and sperm out of? Well, a lot of these fish are just residents. They're living here regardless. And remember, the females are only spawning once every 3 to 5 years. Some males will spawn every year, but most are doing every other year. So we have to handle a lot of fish to be able to get what we need to collect eggs. So as we collect fish, you know, we don't waste this. We put a lot of resources into this. We get biological data from everything. So we'll get a length. Every fish gets weighed, and every fish gets a pit tag. And for those who don't know what a pit tag is, it's the exact same thing that goes into your cat or your dog if they've been chipped at the vet. You can run a scanner over it, and you get a unique number that only belongs to that animal. So this is how we track our fish. Well, track is maybe not a good term. We monitor who we have. We've had fish that we've caught, I don't know, 7 or 8 times over the last 20 years. But having these tags in allow for 2 things. If the fish migrate out of our system into other areas, researchers can get the code off of that tag, and we have a database where we share information. So that researcher can contact whoever tagged the fish, get all the basic information out of it, and it helps the the understanding of how these fish move and how they grow. The other thing is we try to use this for responsible genetics. We don't want to take eggs and sperm from the same fish years, well, can't get it year after year, but we want unique fish. Every time we take eggs, we want unique genetics. So this allows us to keep track of who we've taken eggs and sperm from, so we don't duplicate that effort. And that came in handy this year. So we had a couple of males that we had to throw back because we'd already used them. So in order to find a ripe male, we basically lay it on its back and give it a good squeeze. If we get a little bit of milk, a little sperm, that's a ripe male. Most of the fish that we squeeze, we won't get anything out of. So those fish are not ripe and ready to spawn, so they'll get released immediately. And that works great for males. Unfortunately for females, it's a little more complicated. So we've got this thing we call a hypodermic egg extractor. Basically, we need to take an egg biopsy before we make any decisions on whether we're going to use a female or not. So they're big, they're rotund, but it doesn't mean that they're ready to give us good, viable eggs. Back, I don't know, I think I started using this about 2012. Prior to that, we actually did surgical sexing on these fish, where we would make an incision, small incision, get a little biopsy, and then stitch it up. And that took 3 to 5 minutes if you were good. This takes 20 to 30 seconds, and it's a lot less invasive to the fish. So we don't anesthetize the fish. These fish are quite awake and feisty at times. So it takes a few people to keep it in its place, so to speak, while we do our procedure. And if you're wondering, okay, are you hurting the fish? It's very interesting, and you'll have to take my word for it if you want. But the belly of the fish, because it's constantly on the bottom, they seem to have very few nerve receptors in that belly skin. So literally the fish doesn't flinch. It makes no movement at all. As far as it's concerned, you're just poking something. Other places, the fish is very sensitive, and then the skin and the musculature will react. So I really don't feel too bad about this. This heals up within a day. So I don't know if you can see the arrow here, but those black eggs there, that's what we're looking for. But just because we get black eggs, it doesn't mean that we're there yet. Ideally, we're looking for, something nice and black. Not to get too much into the weeds, but there's different stages of black. There's stage 4 and 5. We're looking for 5. If they're 4, we can't tell the difference just looking at them. So that's where we start in looking. This is an example of immature eggs. These are stage 3 eggs. These eggs wouldn't be ready until the next year. So using the hypodermic extractor also allows us to track females that we might not otherwise even know that they're females. Learn what stage they're at. If they're collected again in the next few years, you might be able to back calculate and see what sequence this female was actually spawning in. So like I said, just because we have black eggs doesn't mean we're ready to go. We have a little mobile laboratory we set up where we hold the fish, and we do this thing called a progesterone assay. So we incubate a series of eggs from each female in progesterone, and we also have a control system, which are just sitting in normal saline solution. They incubate for 18 hours, and after that, they are hard-boiled and cut in half and observed under the microscope. And that's what a hard-boiled section sturgeon egg looks like. And the important thing here, and I'll tell you up front, this is an egg that failed. This egg tells us that the female is not ready to be, the egg's not ready to be fertilized, and we would let this fish go. And what we're looking at is this black dot right here. It's called the germinal vesicle. It's basically the nucleus. That's where the genetic material is encapsulated. what we're looking for is for that nucleus to basically, jumped ahead there, to basically disappear. That way, when the sperm penetrates the eggs, the genetic material can mix instantly. So that's what a bad sturgeon egg looks like, and that fish would be released. We can't hold her and make her get ready. We've tried things like that, so that fish got a ticket out. So once we get there, we'll have 3 to 5 females, the big ones in the upper left, maybe upwards of 20 ripe males in a couple tanks. We now have 2 tanks to separate them so they're not too crowded. Those big red tags allow us to identify the fish just looking down. We don't have to handle it, just turn the water off. We can read the numbers and know who's who. But like I said, we're almost there, but these fish aren't ready to give us eggs and sperm on our timing. So we have to convince them to do it on our time frame. And for that, we have to induce them with hormones. So there's 2 different hormones that we can use, the synthetic LHRH or carp pituitary. We don't use a synthetic anymore because of 2 things. Number 1, it's very expensive. And number 2, the material safety data sheet is about 8 pages long. It's really not materials we want to handle if we don't have to. So we use carp pituitary, which is basically pituitary glands from carp that have been harvested, freeze-dried, and ground up. It's very simple process used all over the world to induce fish to spawn. So fish get injections. Females get 2, males get 1. And from 32 hours after we start this series, the fish are ready to spawn. So this injection of hormone is not going to make the fish spawn. Basically, it's jump-starting the fish's own pituitary gland to produce those gonadotropins to want to spawn on its own. This is just sort of a jump start. So then we finally get to egg take day. First thing we do is we have to harvest the sperm. So if you were dealing with a fish that's a couple pounds, you can hold the fish over a bowl of eggs and everything is fine. But when you're dealing with fish that are 30 to 40 pounds, that's a little more challenging. So we harvest the sperm up front. This fish is in overdrive now, so we'll fill up a 60 cc syringe without any issues. After we harvest the sperm, it goes to the lab and we look at the motility of that. If it's less than 75% motile, we won't use that fish for fertilization. Typically, we'll have about half of the group are 90 to 100% motile, and those are the fish that we'll use. So then we put somebody, after all of the sperm is harvested and we're ready to deal with the females, we'll put somebody in the tank. And then we lift the fish, we let the body weight of the fish push eggs out. Obviously somebody holding the tail, somebody holding the head. That's me holding the bowl of black gold, we refer to it as. So unlike our normal fish, when you take eggs from it, you strip the female, you just push all the eggs out and it's all over. Sturgeon have a very unique internal anatomy. And in the wild, these fish will take 24 to 48 hours to spawn out all of their eggs. They don't just dump them all at once. And same thing here. We can't ask the fish to give us a lot of eggs at once. So we'll go through this process 3 to 4 times with each female to get what we're looking for, a quota of eggs for the hatchery system. So once the eggs are taken, we water activate the sperm. The sperm doesn't do anything until it hits water. So we have to be very careful not to get it wet prior to. It's only good for about 1.5 minutes. So we fertilize our eggs, and there's a few other processes that I'm not going to bother to tell you about because it's just processes. And if everything worked right, this is what a 2-day sturgeon egg looks like in the hatchery system. This is a developing embryo right here. And sturgeon develop incredibly fast in the eggs. In a hatchery situation, if the water happens to be really warm, have had them hatch in 4 to 5 days, which is amazing. In most situations, it's going to be, you know, somewhere between 7 and 14 days till hatching. But even so, that is remarkably fast for any fish species. And then at the end of September, early October, if everything went right, we've got buckets full of handsome little fish to go out into their environment and hopefully survive for many, many years. And finally, Black Lake. What everybody's been waiting for, I think. So, like I said, in the management plan, there's a whole bunch of different management units, and Black Lake falls into the Upper St. Lawrence Management Unit, which is the St. Lawrence River from Cape Vincent all the way to the Moses Saunders Dam on the upstream side, and the Oswegatchie River, you know, running up as far as Natural Dam, and, you know, breaking off on the Indian River side up to Black Lake and Ross Sea. So that is essentially the management unit. We're looking to try to identify 750 adult female, or not female necessarily, adult spawning age fish and evidence of natural reproduction. Now, we've determined some natural reproduction in part of the Oswegatchie River. You know, I think that part of the box is checked. But, you know, we've got, in the St. Lawrence, we've got different counts and numbers of different, you know, different ways. It's not a clean way to count and say we've got 750. So Black Lake was just a piece of that puzzle to see if we could get to that number. So Black Lake is sort of our hub for stocking distribution. Two hatcheries raise our fish. Our Oneida Lake hatchery, which is a walleye hatchery down on Oneida Lake. And we also send fish out to Genoa, Wisconsin, to a U.S. Fish and Wildlife Service hatchery out there. And those guys specialize in sturgeon, and they are very good at raising fish. And what you see here is the Genoa trailer on the left, ready to distribute to one of our DEC stocking trucks to take those fish further up. We stock fish below the dam at Hawkins Point. We stock fish in the lower Racket River. We stock fish in the Salmon River in Franklin County. So that's where our stocking truck is gonna, you know, relay some of those fish. And fish are actually counted out by weight. So you see a scale hanging right there. You know, nobody actually physically counts out 1,000 fish if we're stocking 1,000 fish. Prior to getting into the hatchery, they'll take a subsample and, you know, get a count and by weight. And so they'll know exactly how many pounds of fish to transfer into that other vehicle. So for Black Lake, the fish that are stocked there, some will go in right at the launch, or if time's not a crunch and it's a beautiful day, you might go up to Ross Sea at the base of the rapids there. And I mean, what a great place to just be for an hour to let fish go and watch them acclimate in the current. It's pretty cool. So that's what we're releasing, you know, anywhere from 5 to 8 inches long, handsome little fish, chocolate brown with black blotches and little bony scutes to protect themselves. And between 1995 and last year, we've done just shy of 18,000 sturgeon stocked into Black Lake. Now, in the grand scheme of stocking, that doesn't sound like much, but most of the stocking that's done is not raising fish to this level. So these fish have a good head start on things. So that takes us down to what we're trying to do. Last September, I think it was the first 3 weeks of September last year, we started on our mark recapture process to try to come up with a number. How many adult spawning size fish do we have in Black Lake? September was chosen for a specific reason. Hopefully the water temperatures were going to be starting to drop, which unfortunately they didn't. but it's also, you have a natural distribution of the fish. They're not getting ready to spawn. They're not congregated. They're just out randomly distributed around the lake. Now, the gear that we use, those big gill nets, are 8 to 10 feet high. So there's parts of the lake that were just too shallow to set them in, because we could not have what we refer to as negative interactions with the public. When a boat goes through and they hit your net, not only do they really ruin our net, but they also have a bad day when they're trying to unwrap the prop of their boat. And when they have a bad day, we all have a bad day. So some of those areas that you see cross-hatched were sort of out of bounds. We knew we couldn't set gear in there to collect fish safely, with all the navigation on Black Lake. So we broke the lake up into two basins: the upper basin, which you see the sections A, B, C, and D, and the lower, the southern basin, in our numeric 1, 2, 3, and 4. And within each compartment there, 5 nets were set. So we did that the first week, and we collected fish and we marked them. Again, the whole same thing. Every fish got a pit tag. Fish got another separate fin clip to mark that fish so we could identify it in the future. I believe we had about 150 fish the first week. So after the first week, we were done. We had 2 crews working simultaneously, one in the upper basin, one in the lower. In the upper basin, which is where I was mostly, we caught fish, but not nearly as many as in the southern basin of the lake. So after we did our first marking, we gave the fish a rest. We did not come back the next week. We let them rest and redistribute wherever they wanted to be. And then we came back would have been the third week, and repeated it all again, looking for the recaptures. How many original fish did we catch? How many recaptures did we get? And it's a pretty simple formula. It's called a Peterson estimate. It's been around since, I think, the 1890s, with a few variations. It's not complicated. We didn't have time and resources to do a complicated mark recapture study. This is the simplest, the quickest thing we could do, and it was, adequate for our needs. So Black Lake, typical September, I'm guessing, green pea soup. Visibility about 6 or 8 inches. This is a photo of our guys getting gear ready to deploy. And this is one of my favorite pictures I think I've ever taken. It's got three aspects. So the guy in the yellow bibs, he's never seen a sturgeon. He has no idea. He was just borrowed for the day. So he has no clue what's going to happen in a second. The guy in the middle, Dave, he's handled many hundreds of fish with me, been with me doing this for many years. And he can tell by the behavior of the net that, oh, wow, are we in trouble? And the guy holding the net, he just thinks, oh, we got a fish on. He thinks that's pretty cool. That was a pretty rigorous net to pull in. And that's what we got. I think that was half of the fish. The big fish there is, I think it's 80 some pounds. That's a 150-gallon cattle watering trough. There's another 4 or 5 fish underneath that. But that's just a taste of what we were in for, on our first days of netting out on Black Lake. And we really did not expect that. So, when you're pulling in a little fish, like on the left here, that weighs a few ounces, not a big deal. But then when you pull in a fish that weighs 150.88 pounds, that begins to be a bit challenging, especially for old guys like me with bad backs. And that really set the stage for what we were in for. We did not catch small fish. Everything we caught was big. And just to jump ahead, over that 2-week period of netting, we handled 325 adult sturgeon. The total weight was 8.68 tons of sturgeon between 2 crews over that 2-week netting period. The other boat was a much smaller boat, and as you notice, the guys in the back have got their hands on our life jackets, because The potential for swimming with the sturgeon was very real. It took a lot of effort to horse these fish into the boat, and then all you do is you look back down the net and you see more coming. So these nets were 300 feet long, and pretty much from the beginning of the net to the end of the net, you had to pull sturgeon out of them. And they did trash our nets. We've never had so much gear absolutely destroyed by these fish. We had the week 2 was sort of an emergency build new gear for our technicians. And again, that's what we ended up with, a tub of fish, and none of those fish are small. So we do our best to weigh the fish, but these fish actually broke some of our scales. And this is the Fish and Wildlife Service crew, Scott Schluter and Justin Ekret, doing a girth measurement. There's some really good tables, growth tables for sturgeon, very accurate to give us a good estimate of how much these fish weigh just by getting the maximum girth and the total length. So I think that was a 115-pound fish right there. So where did we catch most of our fish? Well, as I alluded to earlier, most of our fish, were taken down in the lower basin down here. The purple dots and the blue dots, if that's how they're showing up on your machines, wherever you are, they're showing you very high densities of fish. That's where those nets, from one end to the other, those nets are loaded with fish. Green dots are 6 to 12 fish captured in those nets. Black dots 1 to 5. So if you just look at the southern basin, which is a little bit different, it tends to be a little deeper. There's some deeper troughs, deeper water, rockier shorelines down through here. You do have the Indian River, which, granted, a lot of times of the year there's not an awful lot of flow coming down there, but that's getting you your clean water flow into the system. So as you go into the upper basin, fewer fish. They're still there. They're still big. We didn't get very far beyond that first island here past the causeway, just because it was so busy. We just kind of left our gear there. But we caught a lot of fish, 325 fish over 2 weeks. A lot of recaptures for sure. But we still kept catching new fish, which was amazing. So just to give you an idea of what the length of these fish were, the smallest fish was in the 40-inch range. The largest fish, upwards of, 75 inches. But the vast majority of fish were all 55 to 60 inches. Remember, 60 inches is 5 feet long. That's a lot of big fish. And I apologize, this table is kind of busy. I had all kinds of other things on here before, but I decided just to boil it down and talk about this table. So again, we caught most of our fish, and I'm hoping people can see my little arrow moving, in the lower basin, 1, 2, 3, and 4. 93 fish in the first unit over 2 weeks. That's amazing. A lot fewer fish. Well, it really comes down to about half in the upper basin. Average length, if you look down this whole column, the average length is just like you saw on the histogram a second ago, like 55 to 60 inches, the average length in each unit. The smallest fish weight-wise was 15.5 pounds. Our largest fish, 150.88. And actually, we don't know if that's an accurate weight, because the way we had the scale hung by a steel bar, the bar was bending, and our people were afraid that if it broke, somebody was going to get hurt. So we took the first number we could get off of it. It may have actually been a little heavier than that. But all those fish, for maximum weights, other than that one unit right there where we only caught 2 fish, but look, 100 pounds, 124 pounds, You know, these are weights of fish that we're not used to handling in the St. Lawrence or in Lake Ontario. We don't see fish this big. And just to try to give you an illustration, we said that our Black Lake fish had shoulders on them. You know, this on the left side is a Black Lake fish. You can see how rotund and round it is. It's just full of meat. It's huge. Versus, you know, a fish taken in Lake Ontario on the right. Granted, it's a lot shorter, but it's more streamlined. It doesn't have that girthy look to it. It looks more normally proportioned. Back to the numbers. So what numbers did we get? Did we get our 750 we were hoping for? Well, no, we didn't. So like I said, the Peterson estimate is sort of, you know, the baseline. It was the first, you know, real population estimate model out there. But a few different variations of it have happened since then. And we use the Chapman version, which is the least biased. Will give you the most accurate range of fish. So what we came up with was 488 and a half fish, you know, as an estimate. We use confidence intervals, you know, the 95%, really to gauge how variable that number is. You know, we know it's not exactly that. We absolutely know that, but the number, you know, is somewhere between about 380 and almost 600 would be a reasonable estimate for Black Lake. So we didn't make 750 in Black Lake proper, but and it's not my job to check the box per se, but you know, within the whole management unit, many of us believe that we can check that box. We had that 750 fish. But moving on to something that every one of us found interesting and were probably a little creeped out by was the lampreys. Remember, early on I showed you the lamprey on where it had attached to the fish and the fish rubbed it off. Well, this here on the left side is a silver lamprey, and that's what's attacking our fish in Black Lake, and at a rate that we had half a dozen fisheries professionals on these boats, and no one had ever seen anything like it. The silver lamprey is actually a native fish. It's our only native parasitic lamprey. They get to be, you know, at maturity 16 to 18 inches long. Can they really do a lot of damage to a fish this big? Probably not, but let's face it, that's creepy. There's a lot of wounding on our Black Lake fish. On the upper right are sea lampreys. Now, sea lampreys are the fish that have devastated the Great Lakes. You know, basically the lake trout have been very susceptible to sea lamprey. And there's a very expensive and long-running control program to try to keep sea lampreys numbers down. So the sea lampreys did not come from Black Lake, but this is just an example to show you the difference between the two. And on the lower picture are the sea lampreys with a silver lamprey for comparison, a much smaller fish. So if you ever have an opportunity to actually tell the two apart, if you ever see one or, you know, handle a fish with a lamprey on it, the sea lampreys have two distinct dorsal fins with a distinct notch in between them, whereas the silver lamprey has one continuous dorsal fin. That's the easiest way to tell the two apart, especially if the sea lampreys are much smaller than these mature adults. So looking at the dentition, they're exactly this, well, they're not exactly the same, but they're very similar, just size makes a difference. On the upper right is sea lamprey, a suction disc to suck onto the fish, very sharp little teeth to help hook and grab on, and that tooth right there actually scrapes through the skin and the musculature of the fish so the fish can feed. The silver lamprey on the other side, on the left, much smaller, but all the parts are pretty much the same. You know, it sucks on, uses those other smaller holding teeth for stability, and then grinds away with that central tooth into the fish. So really pretty creepy. Again, looking at one of your fish straight down, there's a mark, fresh mark, another one. Let's see, 1, 2, 3, 4, 5 very fresh marks. Those lamprey probably fell off as the fish was in the net. And our big fish that we had, our 151 pounder, there's 7 marks that you can see. There's some healed marks, and on the other side of the fish there was more. So these fish were really just getting hammered by lamprey. It was really amazing, and like I said, none of us had ever seen anything like it before. Another little side note here, this is pretty simple. I'll just tell you what things are. If you can see the red line here is basically each end of our basin where we netted. And we had fish that we collected here that were recaptured 2 weeks later down at the other end. So these fish were constantly moving back and forth and around. So they didn't have a really tight home range. They moved wherever they wanted to in the lake. The green squares with the black line is an illustration of, in 1998, a juvenile sturgeon that was stocked. I think it was 2 years old when it was collected and tagged. It was tagged in the Oswegatchie River up there, and we recaptured it down here in Black Lake. So that's just an illustration that these fish move in and out of the system. We've had fish tagged in the Oswegatchie below Eel Weir that have shown up below the Moses Saunders Power Dam in our spawning collections. So moving downstream past these dams, not a big problem for the fish. They can get around them. Obviously getting back up has been the challenge. So these fish move quite a bit. So in parting, we're about done here. That's an example of a 100-pound plus fish, Black Lake Classic. The tip of the dorsal fin has been cut off as one of our marks. So if somebody happens to see one with the stubby little dorsal fin, you know that he's been handled by the DEC. This was, for those that Heidi didn't say, I'll be retired by this time next week. And this was a great field event to finish off with. It was amazing to handle these fish. They are just so wicked cool. They've come such a long way in restoration, and hopefully they will continue on, and in the next few years we'll take them off of our threatened species list. So we're the New York Department of Environmental Conservation, but none of this would have happened without the help from the Fish and Wildlife Service, our folks in the Cortland office, Scott and Justin, the Genoa National Fish Hatchery led by Doug Aloisi. Amazing folks to work with. I just can't say enough about how much they have helped the restoration process. And not only with sturgeon, but with other species. The Genoa Hatchery actually does a lot with freshwater mussels that are endangered. They've got quite a repertoire of expertise out there with a lot of different things. So I guess with that, I'm going to say that I'm done.
Wow, thank you. Quite amazing projects you've done, and quite a career, so congratulations on your retirement. We did have some questions come in the chat box. So I guess I'll start there while everyone else is sort of thinking if they want to ask anything. If you want, you can unshare your screen. Oh, I got it. So Linda was asking about the photo of the lab in the very beginning and wondering if that is the PATH lab at Cornell.
No, that's a Fish and Wildlife Service utility trailer that gets absconded every year for about 2 weeks. They throw everything out of it, drive it up to the NIPA property where they give us a secure location. for our tanks, and they've got security watching over the fish when we can't be there. So that trailer sits up there, and it's used just for that purpose, just to evaluate eggs and evaluate sperm and store a little bit of gear in case it starts to rain.
Well, also, Gretchen was wondering if you know which type of sturgeon is on the Columbia River in the Washington, Oregon area.
Those would be white sturgeon, which is sort of the alternate of our Atlantic. You know, the white sturgeon again can be upwards of 1,000 pounds or more, and they've run through all of the same problems that any sturgeon species has: impoundment by dams, moving them around, harvest. So yeah, every sturgeon species has the same issues, but the Columbia fish, those are the big guys.
And we have a high school teacher who is interested in career paths, and if there's any advice you could offer in the way of career paths for students that might not want to go to college.
In the government systems, I would say that you're going to be required to do some future study in universities. You know, whether you could get away with a bachelor's degree, most people will have a master's, at least in New York. Not everybody, for sure, but boy, that would be difficult, because even our technical staff, you know, those who aren't, you know, regular biologists, but you know, the people who do the real work, by and large, the real grunt work, you know, even those folks are generally going to need at least some college education. I don't have a lot of good news in that vein, I guess.
Okay. Sue, did Sue have a question? Did you want to unmute yourself, Sue, and ask a question? I see she has her hand up. I thought she might have a question unless she mistakenly hit that button. Let me try and unmute you. Not able to get her, so maybe it was a mistake. If not, if we can't get to you, you can use the chat box and I'll say it out loud for everyone. We have questions. In the DEC, how much time do you spend in the field? Are you out working in the waterways most of the time, or do you have office time too?
Speaking for myself, it would be about half and half. You know, our field season would normally start beginning of April and go through September, maybe into October. It depends on what projects you have. We do have some brook trout egg take things going on in late October, early November. But for me personally, it was pretty much 6 months in the field and 6 months in the office.
And Alden is wondering, what is the best way to treat a fish if you were to accidentally hook one while you're fishing?
The absolute best way is to leave it in the water and try to get the hook out without taking it out of the water. You know, now granted, you saw us taking pictures with us with those fish. Those fish had been through a lot. And we go through great pains to handle them carefully, even when they're out of the water. But ideally, people should not, you know, take them out of the water. Sometimes you have to get the hook out. I'd rather, if the hook is visible and you can get to it, I'd much rather have the hook come out than cut the line. But basically, you know, just like any fish, the more you handle it, the more slime you could remove, leaving it susceptible to fungal infections and things. So, you know, for the best case for any fish is, you know, leave it in the water as much as you can.
We have another species question. Virginia was wondering about sturgeon in the northern lakes of Wisconsin, because her grandfather always talked about catching sturgeon in the Illinois River.
Yeah, well, I mean, those would be lake sturgeon, and Wisconsin and Minnesota are probably I would say, as far as in the United States, the leaders in sturgeon biology, moving ahead with restoration efforts. Those are the folks that have come up with the nature like fish ways, and they've been doing this a lot longer than we have. So I would give them the credit of being, ahead of the curve. But yeah, there's a lot of those upper Midwestern waters that still have some fairly good populations of sturgeon. And people can catch them pretty routinely.
Another question from Linda. She was wondering, how do you tell the age of a sturgeon?
Well, sturgeon, unlike, they don't have scales. Most of the fish that we age by scales. It's like counting the rings on a tree. Another method, if the fish is sacrificed, is to take the otolith, which is the ear bone. But sturgeon don't have good otoliths, so bonus for them. And we're not going to sacrifice a fish just to get the age. So we actually clip off the leading edge of the pectoral spine, which is his front spine right up here. We can cut that out. The fish will actually, it'll heal up pretty well, but then that can be sectioned and looked under the microscope, and that will lay out lines similar as the tree. So you can just count them up.
Great. Does anyone have any other questions? Anybody want to unmute yourself and ask a question? Feel free to.
I see one pop up from Dan. How do we tell a stocked fish from a native fish? That's not a perfect, I don't have a perfect answer for that. We go through a lot of gyrations trying to figure that out. And when you're stocking out, 20,000 fish statewide, it's hard to mark them all. We've tried chemical markers like tetracycline, which you need to take a sample of the fin ray and look at it under ultraviolet light, look for a mark. That didn't work out very well. We've tried scute marking, which is when they're in the hatchery actually taking off a couple little scutes in certain combinations. But as the fish grows, the scutes, a little bit like teeth in a person, if you got a gap, they might migrate a little bit. So it's really hard to count. Now what we do is we have coated wire tagging. So a microscopic, you can't hardly see it, little wire tag is injected in the snout of our stocked fish. And we have these readers, which are basically magnetometers that we take out with us so that we can just basically wand a fish. And if it beeps, it's like, all right, that's a stocked fish. If it doesn't beep, and doesn't have anything else, any other missing parts, anything that let us believe it's anything that a natural reproduction, that's how we do it.
And
I have a question. This is Will. Hi, Roger.
Hi.
You haven't seen me for a long time, but down in Houston.
Yep.
I asked two questions in one, and that the second one was a comment that my grandfather, probably in the early 1900s, up through maybe 1915 or something, talked about getting sturgeon out of the Illinois River. Since then, it got way polluted. Now it's much cleaner. At what point would sturgeon come back, and what sturgeon are those?
Well, there's still lake sturgeon. Those are the only fish we have in the Great Lakes basin. At what point? Well, basically, there's some folks that are going to tell you, and I don't know if it's a proven thing, that fish will home back to their native stocking or, hatching sites similar to what salmon do. So in a lot of cases, when places, different states are trying to resurrect sturgeon populations, they may be rearing them actually streamside to use the specific water right from that area, and then do some habitat improvement to create some spawning ground. You do that and you hope that in 20 or 30 years after you've been retired that something actually happens. There's areas of the upper Great Lakes are doing some really amazing restoration projects right now. Some very big projects right in major shipping channels in the Detroit River and such. So there's actually a lot going on in the upper lakes. And I'm sure a lot of that information you can tap into. But yeah, the best thing that ever happened to sturgeon was the Clean Water Act in 1973. That is the best thing that ever happened to them, because then we stopped dumping junk in the water, or at least we keep keeping track of it. So things are getting better. Fish are coming back. It just takes a long time.
Thank you. Congratulations. Nice talk.
Thank you.
And another question about, are there sturgeon in any of the other Indian River lakes like Butterfield?
No. Black Lake is the only one that we've stocked basically because of that connectivity with the Oswegatchie. And there were historical reports. I mean, historically, these fish were in most of our major tributaries to Lake Ontario and the St. Lawrence River. And, over time, with dams, they just got lost. Interestingly enough, the Grass River, there's a remnant population between Madrid and Messina that has been hanging on there for a couple hundred years, and they're doing okay between those two impoundments, and they've been doing okay.
Anybody else have any questions? Yes. Well, hearing none, I'll thank you again, Rodger, so much for your last presentation of your career.
Yep.
And thank you all for joining us tonight for the Summer Science Series of the Indian River Lakes Conservancy. We hope that you can join us for Cape Cleary coming up on August 13th. We're always on Thursday nights at 6 p.m. So have a great night, everyone. Thanks.
Okay.
Thank you, Rodger. Good luck in your retirement. Great presentation.
Thank you.
All right. Take care. Stay in touch.
Yeah.
All right.